Hello everyone, my name is Dami, and I'm going to take you along with me as I do a DNA extraction in my lab today. So I have these mouse ears that were clipped by my wonderful lab manager, Maria. This is what they look like in the Eppendorf tube, and the first thing I'm going to do is organize them by their number. Essentially, once the pups are born, the ears are clipped in a certain position that corresponds to a certain number. So the clipping allows us to identify the mouse that we need when we need them at a later point in time. So this is what it looks like when everything has been fully organized, and the next thing I'm going to do is some calculations, because the first step of the DNA extraction is to make an aliquot solution for my 14 samples. So you can see here that I need 7 milliliters total of TNES buffer and 70 microliters of proteinase K. And so once I have that all calculated out, I'm gonna heat up the TNES buffer in the microwave, because as you can see, it kind of crystallizes once it's been out for some time. And so as you can see here, after putting it in the microwave for a minute or so, the solution is pretty clear. And now I'm also gonna get the proteinase K, which is stored in the fridge. So I'm gonna get that out of this box here. You can see me kind of struggling to do it with one hand. And once I have that, I'm going to use the electronic pipette to pipette up 7 milliliters of the TNES buffer. TNES buffer is made up of SDS surfactant, which breaks down the cell membranes in the tissue, tris buffer, which will keep the DNA at an optimized pH, EDTA, which will inhibit the nucleases that break down DNA, and NaCl, which will destabilize other proteins and neutralize the charge of the DNA, which will help it precipitate out later in the experiment. So next I'll use this micro pipette to get 70 microliters of proteinase K into my solution. What proteinase K does is it digests proteins, including histones and other structural proteins, which will help the DNA be released from the cell. And once I have all of that inside of my tube, I'm going to go over to the vortex, give it a nice spin just to make sure that everything is mixed up and dispersed evenly. So now I'm going to put on gloves, which I definitely should have done before I started the whole thing, but I digress. And yeah, get all the tubes open. And yeah, now I'm going to pipette 505 microliters of my solution into each of the tubes with the ears. Here, you can see me doing that. Wonderful, great job. Yes, I'm going to give it a good shake just to make sure that the ear is completely submerged into the liquid and not like hanging on the side or something. Yeah, it should look something like this. And I'm going to go ahead and put this in the 55-degree incubator and leave it overnight. And hopefully everything is nice and dissolved when we get back. And yeah, so now it's the next day. I've gone to get my ears out of the incubator. They look nice and wonderful. Here is a closer view of like what a dissolved ear looks like. You can see it best in this next one that I'm about to pick up. Yeah, you can kind of see the gunk at the bottom and then the liquid at the top. That's what you want to see. And now I'm going to pipette 150 microliters of saturated NaCl into each tube. What the NaCl is going to do here is it's going to cause all the proteins to precipitate out of the solution, which is going to let us separate out the proteins and all the other gunk from the DNA and from the liquid in the solution. And so now I'm giving it a good 30-second shake, and I'm going to let them incubate in the minus 17 degrees Celsius freezer for 17 minutes. And what you want to see is a cloudy solution, which is a sign of the proteins precipitating out of the solution. And what I'm doing here is I'm putting them in the centrifuge, and it's important to make sure that the centrifuge is balanced perfectly so that as it spins super fast, it doesn't explode. So I'm putting the cap on, putting the lid on, and then I'll let it spin for 5 minutes at full speed. And what that's going to do is it's going to use centrifugal force to sediment the aggregated proteins and the cellular debris. It'll make a pellet, which you can hopefully see here. Yeah, you can see here the pellet with the gunk and all the protein and the supernatant, which is the liquid at the top. And so now I'm just gonna make new tubes because we're about to transfer the supernatant into a new tube. And again, they're numbered. I'm gonna get 500 microliters of the supernatant and put it into the new tube. The supernatant contains the DNA, so that's why you want to separate it out from the gunk and all the protein at the bottom. And you can see here the new tube on the left and the old tube on the right. What you're about to see me do here is give it a like firm but not harsh shake. That's just going to make sure that everything gets mixed up but without disturbing the DNA. And we're adding ethanol because DNA is not soluble in ethanol when salt is present. So since we added the salt earlier and we just added the ethanol, the DNA is going to aggregate and precipitate out of the solution, forming the white fibrous material you can kind of see floating around here. Depending on the sample, you may not see the DNA visibly with your own eyes, but you kind of just have to trust that it's there and then put it back in the centrifuge. Here it's super important to make sure that all of your Eppendorf tubes are facing the exact same direction because the DNA pellet that's going to come out is going to be super small. And we're going to suck out all of the supernatant, and you don't want to accidentally suck up your DNA pellet. And I'm going to let that spin for 15 minutes at full speed, and here's what that looks like. Now, like I was saying earlier, we want the pellet instead of supernatant, and yeah, you literally can't even see the pellet here. But since we have oriented the tubes in the same way, we know it's going to be at the back just based off of the physical principles. So now we're going to suction up all of the ethanol and other liquid that's in there. You can see me doing it close up here. That's a great angle. Yeah, it's important to not mess up the DNA pellet because obviously we need the DNA for our future experiments. And I'm just going to do that for all 14 of my tubes and leave it out overnight to dry. Yeah, this is what it looks like. So pretty, so awesome. We love science. Now it's the next day, as you can see from my new sweater, and I'm gonna get 100 microliters of TE buffer and put that into the DNA pellet. TE buffer is essentially just a buffer solution that'll keep the DNA at a super stable pH and prevent any of the leftover nucleases from breaking down the DNA. You really want to get in there with the pellet. You can see me, I'm going to show another close-up here. I'm going to pipette up and down and also like scrape the DNA pellet off the bottom of the tube. Since it's been drying out overnight, it's like pretty solidified at the bottom there. Yeah, you really want to get in there, pipette up and down, swirl it around just to make sure that the DNA is resuspended in the solution. And I'm just going to redo this last step for all my 14 tubes. This is what it'll look like. And so now we have DNA that we can use for our PCR and genotyping experiments later.